Good afternoon, and welcome to UCF Coastal's Lunch on the Coast Speaker Series. I'm Dr. Graham Worthy, Director of UCF Coastal. Through this speaker series, I hope you gain appreciation for the extent of basic and applied coastal research underway here at UCF. Orlando may be an inland city, but we still contribute to the issues facing our coast, and are in turn impacted by those same issues. One of the goals of UCF Coastal is to grow public awareness about the amazing coastal resources we frequently take for granted. To achieve that goal, we've assembled an incredible team of cutting-edge researchers who serve as a resource for everyone from marine scientists to the general public, businesses, and policymakers. Our Lunch on the Coast speaker series is running from July through December, with one speaker each month. Through this series, we're proud to highlight some of the recent research undertaken by the diverse interdisciplinary faculty of UCF Coastal. You'll learn what it really means to live in a coastal state, with expert perspectives ranging from archaeology to coastal planning, from engineering and economics to ecosystem and human health. You'll get an appreciation for the full range of issues currently facing our coasts and the resources that UCF Coastal is bringing to bear to help foster an understanding of those threats and hopefully chart a path forward. Now let me pass you over to our moderator, who will be introducing you to this month's speaker, and we'll also be running our question and answer session following the talk. Thank you for joining us today, and we hope you enjoy the Lunch on the Coast speaker series.
Hey everybody, I'm Chris Emrick, UCF Coastal's Director of Research, and I'm here today as your moderator for our Lunch on the Coast speaker series. It is my great honor to introduce to you today's speaker, Dr. Michelle Gaither. Dr. Gaither is an assistant professor in UCF's Department of Biology and also holds a secondary joint appointment here with UCF Coastal. Dr. Gaither's particular research interests and expertise in implementing molecular tools to understand how geography, ecology, and selection influence biological diversification are critical to a broader understanding of the processes occurring in our coastal communities. Today, Dr. Gaither will share with us some results of a recent statewide environmental DNA or eDNA survey of our nearshore areas that will help society grow in our understanding of marine communities. eDNA is emerging as a useful tool not only for scientists like Dr. Gaither, but also stands to provide more and better data for coastal conservation and coastal resource management. This short video provides just a glimpse into Dr. Gaither's current research on coastal systems.
For people working in terrestrial systems, it could be fairly easy to go out and count the number of moose on a, or deer on a pasture, let's say. For marine or aquatic systems, you actually have to put, we don't live in those systems, right? We're literally fish out of the water. So we have to get into those systems and collect those data via other tools. My name is Michelle Gaither and I study eDNA and evolution in fishes. eDNA is short for environmental DNA, and it basically is the DNA that all of us shed continuously. Fishes that swim through the water also shed cells, they shed mucus. When they reproduce, they shed gametes into the water column. When they die, they shed, their bodies and their decomposing bodies shed DNA. And that DNA that's found in the water is what we call environmental DNA. First thing you need to understand in order to conserve a resource is what that resource is, where it exists, and when it lives in that or exists in that place. So that's just the baseline data for any kind of conservation efforts, and environmental DNA helps us to get at that. Environmental DNA has as a tool, has the ability to help us understand better how communities change, which species live where, and when, and how that will change through time. Marine or any kind of aquatic realm, it's much harder to get a sense of who lives where when. I mean, if you're just on your boat, you don't know who's below. And so eDNA is one survey tool that can help us answer those questions in a much more efficient and hopefully a much more complete manner.
Should you have any questions or comments during Dr. Gaither's presentation, please use the Q&A button located at the bottom of the screen. Here you can ask questions that we will cover during the later part of the lunch session. Thanks for your questions, and thank you again, Dr. Gaither, for taking the time to meet with us today. The floor is all yours.
Introduction. Okay, so today I first thank you for the introduction. My research program here at UCF is really divided into two main areas of focus. So about half my students are focused on looking at population genomics of marine fishes specifically, and the other half of my students tend to work in environmental DNA, which is what we're going to talk about today. So biodiversity is important, right? Species and ecosystems provide essential goods and services upon which human beings and our well-being depends. Those systems support our health, our environment, and our economies. And just to sort of put that into some perspective and to give you just a glimpse at some of the goods and services provided by our natural systems, we can look at just data in Florida and look at recreational fishing, for instance, or fishing. So recreational fishing in the state of Florida supports about 115,000 jobs. The commercial industry supports another 93,000 jobs, and together they generate about $18 billion in sales and another $3 billion in income. Now, human impacts on our natural systems have been profound. And let's just consider the impact of fish on fishing, or fishing on fish populations, for instance. So as humans became more efficient at harvesting fish, right, we've depleted the most desirable fishes in those nearshore stocks. And as a result, fishers have had to move offshore. Technologies then allowed us to extract resources more efficiently, right, and allowed us to move into deeper waters to extract resources. And as a result, there's been a massive depletion of traditional fishery species. So in 1998, Daniel Pauly, who's a very well-known ecologist, published this concept of what he called fishing down the marine food chain, or fishing down marine food webs. And his idea was based on the premise that the most desirable species, those with the greatest commercial value, are the species at the highest trophic levels. Now, think about cod, right, or tuna, or swordfish. And that these species were the ones that were first depleted. And as we've depleted those, we've moved down the food web, right, and started to fish alternative sources or alternative species. Now, since then, that was published about 20 years ago, and since then there's been more data collected and a reevaluation of the data. And it turns out that Daniel Pauly's idea was a bit of an oversimplification of a really complex situation. But some of the ideas hold up. First, that traditional fisheries, as they began to decline, right, fishing fleets evolved and developed to exploit less accessible populations. So they had to move offshore, or they moved into deeper waters. And that was followed by shifts to less desirable species and more marginal fishing grounds. So let's look at an example. So these are slime heads, or the slime head, right? Now, clearly based on this name, nobody had ever considered that this would be a commercially valuable species, right? It's found in deep waters. It was hard to access. It's found 400 to 900 meters, so it's pretty deep. But it turns out that this fish has a very nice flaky fillet, so it's quite nice to eat. So with some clever remarketing, it's now known as the orange roughy, folks started to utilize this resource, or started to fish this fish in the 1980s. The production peaked in the 1990s and then basically collapsed afterwards. There was a swift decline as we overexploited this particular resource. It turns out that we've done this not just with orange roughy, but with many species of deep sea fishes. So these are not traditional species we would have fished, but as fishers were trying to find new fisheries, new resources, they began to fish deeper. So here's some examples. This data comes from Canada, the Atlantic coast of Canada. So these are all, what do these deep sea fishes have in common? They're all long-lived and they're slow-growing. An example, orange roughy might be kind of extreme, but there are individuals of orange roughy that have been recorded to be 150 years old. They have a long generation time. For instance, orange roughy doesn't reach sexual reproduction or sexual maturity until 30 to 40 years of age. That means it has to live for a full 3 decades before it can ever reproduce. They aggregate on topographic features like seamounts, so they're easy to harvest, and they have sporadic recruitment. Together, these shared characteristics mean that these deep sea resources are highly vulnerable to overfishing. Now, we don't have to go to Canada and look for declining fisheries, right? We can look right here in Florida. This is a paper that was published back in 2009 by Lauren McClinkin. So she wanted to look at the change of fisheries at Key West. Now, official fishery data hadn't really wasn't collected until the 1980s, but she wanted to go further back in time. So she decided to use historical photos, right? So here's a photo from 1957, 1980, and 2007, right? It doesn't take a statistician to figure out that there are some massive changes in catch over that time. She compiled these data and found, here's data on bony fish and sharks. You have 1956 to 1960 time period to 2007, and mean size of the catch, basically data that she was able to derive from those photos. And she found that there was a 50% reduction in fish size, okay? But it wasn't just the size of the fish that changed, but the composition of the catch as well. So here we go with a list of species, starting with sharks at the top, Epinephrus or groupers, cobia, barracuda, et cetera, down the list. You start 1956 to 60, you can see that there are some big sharks, a significant number, this will be the most surprising to folks who are familiar with fishing now, is the number of big groupers that she recorded, 25% of the catch. Now, remember, these are all from Key West, so these are off of head boats. So these would have been, you know how they have the trophy boards down at Key West? That's exactly where these photographs were taken. So this is the kind of catch people were catching back in the 50s, in the late 50s. Okay, so big fish, 1965 to 1979, you start to see a shift, but still lots of big fish. Big fish, by the way, the black bars mean greater than 100 centimeters. The gray bars are between 50 and 100 centimeters. And those white bars are less than 50 centimeters, so smaller fish. By 1980s, those Epinephrus, those groupers are gone. There's no records of them in the pictures that she was able to recover. 2007, so you see a significant shift, right? You're already missing certain taxa. In 2007, you still have sharks, but notice they're no longer the big sharks. The groupers, there's a few groupers, but they're all small, which is that second bar. You have some good size cobia, but really the bulk of the catch, 45, almost 50% of the catch is that big white bar. Those are small snappers, which is exactly what you see in that photo from 2007. So significant shifts. Now, the goal of resource managers and conservation biologists is the long-term management and maintenance of these resources, right? Not just for use now, but for use in future generations. How do we manage these changing resources? Well, the first thing we need is data. And those data are typically collected in two different types of data, right? Fisheries dependent data, which is actual catch from commercial fishers or from recreational fishers that have to be collected, or fisheries independent data. And that's what we're going to focus on today is really the independent monitoring. These photographs basically represent different types or different ways in which those kinds of data are collected, right? You have to be able to, these are typically staff, they're FWC staff, right? Florida Fish and Wildlife Conservation Commission who do these surveys. These would be FWC staff. They might be NOAA staff, they might be research biologists, but they have to go out in the field. They have to put a net in the water, traps, electrofishing if you're working in freshwater, or scuba if you're working on coral reefs, and you have to actually visually observe or physically catch these fishes in order to mark their presence, right? In the IRL, the Indian River Lagoon alone, FWC conducts 1,200 seines and trawls per year, right? That's just in the IRL, and FWC does these surveys all around the state. So we're talking about tens of thousands of people hours per year in order to collect the data that we need for proper management. And this is where eDNA comes in. So eDNA is a potential tool to help augment or even potentially down the line replace some of this survey effort, okay? Now, what is eDNA? eDNA broadly defined is, first of all, it stands for environmental DNA. And when we're talking about eDNA, we're talking about the genetic material that is shed or excreted by organisms. It can be skin, hair, urine, feces, mucus, or it can be gametes, right? A lot of fishes are broadcast spawners, so they release sperm and egg into the water column. that can be environmental DNA, right? And because, of course, I work in either marine or estuarine systems, my environment is water. Now, when I define eDNA, I'm also talking about whole microscopic organisms that I might collect in a water sample. So we're talking about dinoflagellates that cause harmful algal blooms, for instance, or zooplankton, phytoplankton, bacteria, etc. Now, how do I collect that DNA? We go out and we collect water samples. They can be surface samples, or you can drop bottles down to collect deeper water. You can also send divers into the water to do that. Once you have your water sample, you then filter it. And on that, you retain the filter, and on that filter, theoretically, is all the DNA that was present in that water sample. That water sample may be 500 mils or a liter of water. That filter now is what you save and preserve. Later on, you come back and you cut that filter up into little bits, little pieces, and you can extract the DNA from that sample using a commercial extraction kit. All right, so the next step is analyses, and the types of data analysis that you use depends upon your question. We can use a single species detection approach, or we can use metabarcoding. I'll talk about both. So for single species approaches, now this approach is based on the design of species-specific primers that will amplify a single taxa, whatever that taxa of interest is. Could be sawfish, it could be groupers, it could be an invasive species like bullfrogs, what have you. And you design a primer set that will amplify that species of interest at the exclusion of all others. It's usually some kind of qPCR or ddPCR approach. Okay, now let me back up just one moment. So these techniques have been used by microbial biologists long before vertebrate biologists got involved, right? They were sampling soil for bacterial communities, and the only way to do that was to use a molecular approach. The first time these approaches had ever been used on a vertebrate species was looking for invasive American bullfrog in freshwater systems in North America. Now, this paper was published back in 2008. It was really a proof of concept paper. It's been cited over 1,000 times since then, and it just showed that we can use these kinds of approaches to detect vertebrates, okay? Then a couple of years later, there was another paper published on Asian carps, again, another invasive species, this time in the Great Lakes. And this study not only showed that it worked, right, that the molecular approach worked, but it also showed that eDNA was actually more sensitive and cost effective than traditional survey methods, right? Then just the next year is the first metabarcoding study. So the two studies I talked about before, those are single species approaches. They design primers to look specifically for the American bullfrog or for the Asian carp. But here we're using a metabarcoding approach, and this was published out of Denmark in 2012. And here the authors designed an assay to detect multiple species. That's what we can do with metabarcoding, is we're not looking just for one taxa, but we're designing primers, or we're designing an assay that will amplify all the DNA in a water sample from a group of organisms, in this case, fishes. Okay, so this is the first metabarcoding study on marine species. And what they found was they were sampling in a boat harbor and around, and using these approaches, they were able to detect all of the key species shown here that they knew or were known to exist in that water column. So this is the first time metabarcoding had been used in a marine system and had been used successfully. So now in my lab, we've been working on these protocols and optimizing these protocols over the last couple of years. It's actually, because the approach is so new and novel, there's a lot of optimizing that has to happen. First thing we did was we published a review paper. Okay, and this diagram shows just how explosive the field has been in terms of, this is the number of papers published using environmental DNA in marine systems. The brown part of the graph shows single species assays. The metabarcoding is in blue. Clearly, the metabarcoding is the more commonly used approach in marine systems. We've also published a technical review recently looking at protocol development and protocol optimization, and that's really where the focus of my research has been in the last couple of years, is really getting these protocols up and running and optimizing them for work here in our systems in Florida. Now, we talked about single species assays, and we've talked about really freshwater. Notice the systems that I've talked about have been all freshwater systems. Well, there's also assays published for, and especially in the last 10 years or so, assays published for marine species as well. For instance, things that are important ecological species like the river otter, rare and endangered species like sawfish, other important ecosystem critters like the bull sharks. So there's lots of published assays already out there. Our lab has been working on publishing or developing single species assays for lionfish, an invasive species here in the Atlantic, the dwarf seahorse, which can be rare in some habitats, and especially the interest is in monitoring these populations with seagrass declines. That's work that some of the undergraduates here at UCF have been working on with me. We've also been working with FWC to design assays to detect and quantify Carenia brevis, which is the dinoflagellate that causes red tide here in Florida. And we have other work that we're doing with FWC on brown tide on that Atlantic coast and some other work. So that's summarizes sort of our single species work. But most of our work in our lab is really focusing around metabarcoding. And that's because we're interested in being able to describe and characterize entire communities. And again, in our lab, we focus mostly on fishes. So primer design is, again, still important with metabarcoding, but instead of designing these primers to amplify a single taxa at the exclusion of all others, you're designing them to amplify a whole taxonomic group. It involves high throughput sequencing, including the Illumina MySeq. It involves a lot more library preparation and quality control of the data. So metabarcoding is a much more expensive approach, but you garner a lot more data from this kind of approach. So an example of metabarcoding. So this is a study out of New Caledonia. These researchers were focusing on elasmobranchs, specifically sharks. And they compared environmental DNA with two other traditional methods. So UVC are underwater visual census or underwater surveys, and BRUVs down at the bottom in the blue are baited remote underwater video stations. So basically baited cameras, okay? And the eDNA is in red. Now with eDNA, they were able to find or detect 44% more shark species than they were using those other two methods. But the key point here is, with eDNA, you see N equals 22. They were able to detect 44% more shark species, but only with 22 samples, 22 water samples. But if you look at the visual surveys in green, almost 3,000 visual surveys. That's a lot of people hours. And the BRUVs, those beta cameras, they were able to retrieve 9 species with 385 camera drops. So we're talking about significant decrease in the amount of effort required with environmental DNA, and you're still getting 44% more species detected, okay? However, notice there are some species that eDNA didn't pick up, okay? For instance, the tiger shark at the bottom, one of the species of hammerheads, and then a species of nurse shark, which the researchers actually knew that their primer set didn't work with, so they didn't expect to get that species. But the take-home message here is that no single method can fully characterize a community, and that the combination of those traditional methods with eDNA actually gave the best or fullest characterization of the community. Here's another study. This was published back in 2017 that takes more of a tree of life approach. In this data set, these authors use 10 primer sets, right? They're still using metabarcoding. So each one of these primer sets is designed to amplify a broad group. In some cases, all eukaryotes, which is incredibly broad, or all metazoans, which is also really big. They have a primer set that was designed to target fish, another for crustaceans, cephalopods, plants, mammals, Symbiodinium. And then they combine all of those data together to try to get basically a view of the tree of life in that particular location. Now, the problem with this kind of approach, it's great. It's a great approach. However, it's incredibly expensive, as you might imagine. That's 10 assays you have to run on each one of your samples. And the other issue with this is because of the primers are designed so broadly, is that you don't get very good taxonomic resolution. So if you notice 38 phyla, but only 54 species, right? So you have 287 families, and what happens with these data sets is you often have to collapse those sequences, those taxa back to a higher taxonomic level, which is what they've actually had to do here. So they really don't have very good resolution at the species level for most of the taxa that they've detected. It's an interesting approach, but unlikely because of its expense, because of the lack of specific taxonomic resolution. It's unlikely to be applied across many systems. The one interesting thing though, and this goes back, so when we compare different methods like the visual surveys, the BRUBs, and the eDNA, no single method was able to characterize all the diversity. Well, here no single assay can fully characterize a community. So it's always going to be a mixed approach will be necessary depending upon the question. So if we now look at data, this is from Mosquito Lagoon. This is data. So the seine and trawl data came out of Jeff Cook's lab here at UCF with his recently graduated master's student, Dakota Lewis, and my postdoc here helped to collect these data sets. So we're comparing eDNA versus trawl and seine data for fish surveys in the Mosquito Lagoon. Now the trawl and seine data represents 16 trawls and seines combined, conducted back in June of 2018. So our graph shows, or our plot shows, basically a list of species that were detected. 46 species were detected using the trawl and seine data combined, right? 76 species were detected using eDNA. Now, those blank spots in the eDNA column, right? Those represent species that they caught in their nets, but we didn't get, right? And vice versa. Notice there's a very large chunk of species. In fact, let's look at the Venn diagram here. 36 species were common to both methods. 10 species were caught in the seines and the trawls that we didn't detect with eDNA. 40 species were detected with eDNA that we didn't catch. They didn't catch in their nets. It's not too surprising that some species would be missed by the nets. Okay, they're small species. Maybe they live in the substrate. They could be species that can outrun a net or easily frightened away. So it's not too surprising that those methodologies are going to miss some species. eDNA is likely to miss species that only reside for a short time in the water column, especially if they're not particularly abundant. Maybe they swim through and with your water sampling, you're just simply not catching them. So there's always going to be sort of the multi-method approach is always going to be sort of preferred if you want to characterize the whole community. But eDNA did capture more diversity, right? The other advantage of eDNA, potential advantage, is taxonomic resolution in this case. So if we look at family Calerpidae, right, notice missing, so the trawl and seine data are on the right, eDNA, we detected all of these species with eDNA, but we're missing some in the trawl and seine. Specifically, we're missing American shad, which is that top line. Also notice there are 2 species of menhaden. So we got 2 species specifically at that level of taxonomic resolution of eDNA. But in the trawl and seine data, they had to collapse them. And the reason why they have to collapse them is because menhaden are difficult to ID morphologically. You have to bring them back into the lab. You have to do some extra work to try to distinguish them, but still they can be quite hard. And so with the trawl and seine data, they were actually forced to collapse those, whereas we were able to resolve those taxa. So there's another advantage to eDNA. In another study, looking in a different part of the Indian River Lagoon, now we're down by Melbourne. We have a sample site inside the lagoon, which is known as our turbid site, and we have another site outside the Sebastian Inlet, which we call our Clearwater site. And if you've done any water collection inside and outside the lagoon, you know why we call them turbid versus clear. There's a lot of tannins in the water inside the lagoon. There's also often algal, large populations of algae in the water, and not even necessarily during algal blooms. But the water is quite dark and quite turbid. There's also a lot of sediment in the water. So for this experiment, we were testing the impact of water quality, looking at the difference between that turbid and clear water. We were looking at the impact of sampling effort, filter pore size, DNA extraction method on the level of PCR inhibition and basically on our metabarcoding results. We wanted to look at how protocol ultimately affected metabarcoding results. And we wanted to look specifically in our system. So one of the important take-home messages from this study was, one is that extraction method really didn't matter. We tested out 5 different extraction methods. Extraction method does impact PCR inhibition, but you can handle that, you can deal with that. And once that had been dealt with, once the PCR inhibition had been alleviated, there was no impact on metabarcoding results. We also found that filter pore size really didn't have much effect according to the pore sizes we tested. Now, if we look at the data for inside the lagoon, we detected 36 species. Bay anchovy was the most dominant. Sheepheads came in next. We had a species of goby. We had mullets. We had all the list of suspected or of known species in the lagoon, and the data was really consistent. The bottom, just ignore QBT, those abbreviations. Those are basically just the different extraction methods we used. And again, there was really no difference between those extraction methods. Now, if we look outside the lagoon, however, in the Sebastian, outside the Sebastian Inlet, what you might notice is how patchy that data looks. And that is not because of extraction method, but instead what we think is happening is that the DNA is so patchily distributed outside the inlet. So inside the lagoon, we're getting high concentrations of DNA. Outside, we're getting very low concentrations of DNA. Remember that outside the Sebastian Inlet, we're over much deeper water. We have much more water mixing. We have probably less biomass per unit of water. And that DNA is sort of haphazardly distributed within the water column. So our water sampling just simply wasn't adequate enough to capture the diversity. And so we get this kind of this haphazard looking distribution of DNA and species distributions in our samples. We did detect 37 species in total, with the Atlantic threadfin herring being dominant, bay anchovy being second. But the communities between those two locations, not surprisingly, were quite different. So the big take-home message for us, though, was that within the IRL, we get lots of DNA, but we also get lots of PCR inhibitors, okay? Outside the IRL in that clearer water condition, we get far less DNA, we get far less PCR inhibition, and we get a lot more variation across our replicates. And what that means is that if we're going to be sampling in those open ocean environments, that we have to sample more extensively, that the 500 mils or so that we had set up for this experiment is just not adequate because of the variability in the DNA concentrations across those replicates. So the million-dollar question is whether or not we can use environmental DNA to estimate biomass, right? Usually these are species-specific questions. We want to know whether or not our DNA results, our read copy, or our estimation of abundance based on environmental DNA comes even close to the reality of what's biomass is found in the water. Okay, theoretically, you should have, as you increase in biomass in any given species, you should have an increased signal in your environmental DNA. The quick answer is yes, but it's really complicated. So here's an example of a study on walleye in Wisconsin. This is Spear et al. 2021. They were estimating walleye density base using nets and electrofishing, and they have eDNA concentration on the y-axis. There's a very nice correlation between what density estimates using traditional methods and eDNA concentration, okay? R squared 0.81. That's about as good as it gets. And in fact, it's all downhill from there. Here's another example. Here is lake trout in Canada. They estimated trout biomass using gill nets. They compare it with eDNA concentration, and the correlation is much less tight, with an R squared value of 0.45. Then we go to, we look at Japanese eel, and they're estimating eel populations using electrofishing. And they do it in 4 different locations in freshwater there. And again, the correlation is even lower, it's 0.34. So it varies a lot. There's a review paper that came out this year. This is part of a special issue that we published on marine metabarcoding in the journal Environmental DNA that's coming out in January. In this paper, they review 63 published papers that are trying to estimate or trying to look at the correlation between environmental DNA concentration and biomass estimates. And they find that 90% had a positive relationship, right? So it's great. Most studies show a positive relationship between actual density of fish and environmental DNA. However, those correlations really do vary a lot. Now, why is it so complicated? Why is it that there's so much variance across studies? And that's because, well, there's a couple of reasons. eDNA or DNA shedding rates vary. You think about this. DNA shedding rates are going to vary from one individual to another, right? How much DNA is an individual shedding into the water column? And that's going to vary with life stage. It's going to vary with body size. Theoretically, bigger fish should shed more, right? Animal density, you can imagine that shedding rates might be higher in animals that form dense schools. Metabolic rate, the higher the metabolic rate, the higher the shedding rate is that's what the trend is. Higher water temperatures, you tend to shed more. And all of these factors are going to vary, either with life stage or with season. DNA residence times are going to vary as well. Okay, so once the DNA has been shedded into the water, how long does it last? Well, that's going to depend on bacterial activity, because bacteria are going to break down that DNA. UV breaks down DNA, so UV exposure is going to play a role. Water temperature and pH are going to also play roles. It's complicated, and it's likely that every system is going to have its own peculiarities, right? That it's going to have to be, these are going to have to be modeled in each system and potentially by species. So it's complicated. We're looking into this and adding to this research. My student Emily Farrell is working with FWC and is collecting with FWC along with their seine and trawl data to try to get at part of this, to see whether or not we can estimate biomass from some of our eDNA data. We're also interested as a lab is using eDNA as a biogeographic tool. And this is really what I promised to talk about today, but as all good plans, we're still analyzing these data and don't have anything for you today, but soon we will. So this map shows marine ecosystems of North America. And these ecosystems are defined by species distributions. And you can envision each one of these colors sort of represents a slight change in species composition. If you look at Florida, we have three marine ecoregions in Florida. The Clarion Atlantic, which is a temperate climate, right? We have in the south, we have the South Florida, the Bahamian, more of a warm water fauna. And then we have the northern Gulf of Mexico, which has greater fluxes in sea surface temperature, and it's really impacted by the anatomy of the Gulf. So we have three different marine ecoregions in Florida. So in 2020, to sort of go at this and test these, we can consider these to be hypotheses that are worth testing, right? And we can use eDNA to test them, to see whether or not these these marine ecoregions hold up, or to see whether or not, to determine whether or not with eDNA we can actually refine these even more. So in the summer of 2020, when most of us we were all shut down due to COVID, my lab decided to go out and do some field work. And we sampled across the state in 11 different locations, both in estuaries and outside estuaries, wherever we could. In total, we collected about 540 samples. And this is work that these samples have already been worked out. We have the data, but the processing of these data take some time. So Emily Farrell, my Ph.D. student, is leading the work on these BioBlitz data. So stay tuned. Hopefully we'll have these for you soon. And listen, with that, I have to thank a bunch of collaborators, including Jeff Cook, and Dakota Lewis, who offered up, and Linda Walters, who offered up some of that data for us, the trawl and seine data. The folks at FWC, who have been incredibly helpful through this, through everything, and of course my funding agencies. And with that, I think I have time for questions.
Thank you, Dr. Gaither, for such an interesting presentation. I know that I learned a lot. As your resident social scientist here, I'm always super interested in these discussions of coastal species, especially because I love to catch them and sometimes eat them, sustainably, that is. So I've been keeping tabs on the questions, and I will try to put them in order. You don't need to look at the questions. I'm going to deliver them to you. So hopefully we have some more questions coming in. There was two questions by Rob Persons. I think the second one trumps the first one. And the second one is, my original question was asking about the accuracy of metabarcoding when detecting species versus the studies that were done before estimating biomass. So the question was, how was the accuracy of metabarcoding method verified?
So, okay, so typically, how we verify the metabarcoding results is folks typically work in systems that are fairly well studied, just as a proof of concept, right? So, or there's also a lot of mesocosm works. So what we do is we create these mock communities. So there's two different ways. You work in a system where that's already been well sort of established, what we know what to expect, or you can develop mock communities where you can actually take DNA from known species and, you know, in a little Eppendorf tube, create what we call a mock community, as in adding known volumes or amounts of DNA from these various species. You then produce your metabarcoding libraries the way you would normally and see whether or not you're actually able to, you know, to recover all of those species. And then particularly you can do it by, if you know the quantity of DNA for each one of the species, then your read counts should reflect that. If you put them in equal number, you should get equal number of read counts. If you put, you know, 2 times as much of the mullet and only one part snapper, then that should be the ratio of your read counts. So there's ways to test metabarcoding approaches that certainly people have been working on.
Thank you for clarifying on that. We do similar things in GIS where we will effectively remove half of the sample and then see if we can predict it. So yeah, those hopefully, Rob, that answers your question. If not, feel free to add a follow-up question. Anyone else, feel free to add questions. I have a question before we move on to some other questions. Was there anything really surprising? As in, did you find the Loch Ness Monster? Did you find something that you were like, I did not know, no one knew, you know, this existed, or, you know, something super surprising in your work?
You know, so we focus on fish primers because that's the main focus of our lab. But those primers are not perfectly designed, which works for us. So with those, we're able to find mammals. For instance, we find dolphin, we find manatee, we get a lot of human DNA, and that could be in the water, or it could be us contaminating our own samples, which we have a lot of controls in order to prevent that and try to pull out exactly where, if the contamination is coming from us and at what stage of the process it's coming. But we get a lot of unexpected, sometimes you even get things like cow or dog, and that is probably runoff potentially, right, coming in. So you can also get DNA from freshwater systems if you're close enough, and as long as that residence time. So you can get some freshwater species in there and just terrestrial species, birds. People are using water sampling. The degree of success is still, there hasn't been much published on it, but even to like seabirds, to try to document the presence, absence of seabirds, et cetera. You certainly wouldn't want to do any kind of abundance on that, but yeah, there's so the kinds of things that you detect is surprising given what's expected. You know, the tools are designed for a specific group of organisms, but often not restricted solely to those.
That's just so awesome. The next two questions are kind of a transition from the science to action. First question is, in addition to research conducted at UCF, and this may be a question for Graham Worthy or others, has UCF supported conservation laws in the state of Florida?
Ooh, yeah, that is a question for Graham. Yeah, I've been at UCF for 4 years, and so I don't think I'm the best person to address this, but I know that there is certainly a lot of interest amongst the faculty. A lot of faculty in the biology department and elsewhere have really applied research and have taken active roles in changing legislation and rules and regulations. But I think that's about as much as I can say to that.
So that question, unfortunately, was from an anonymous attendee. We will do what we can to more fully answer that question, maybe put out a little briefing on what we're doing, because I also, as the science director, I'm not really super involved in that end of things, but I know what's happening, just don't know to what extent. The second question in the same vein is from Jennifer Chalky. have you been able to implement your research to improve a specific habitat or replenish a species in its habitat?
Yeah, not yet, but that certainly is the long-term goal. So this is true across environmental DNA. So, okay, so environmental DNA really started with freshwater folks and looking for invasive species. In fact, the USGS has massive efforts along these lines. They run fairly sizable government labs that are processing eDNA samples that come in from all over the US looking for invasive species in freshwater systems. It's much more efficient and economical for them to go out and sample water and send the samples in than it is to put people in the water or do electrofishing or whatever other methods they're using for detecting these invasive species. So I know that the freshwater folks are really on the really the work they did was really foundational. The marine folks, we're still figuring out exactly what these tools can do. We know they work. We know that they're excellent at telling us what's in the water. The biomass question is again the $1 million question that will make a big difference if we can figure out that if we can get the models to do that. And so right now, getting all the protocols in place has really been and trying to get some agreement across the community so that we can compare studies done in the IRL with studies done, in Apalachicola or studies done, up north. And that has been, it's a long process. Biomonitoring will be forever changed. And environmental DNA will undoubtedly be incorporated into the way that we do monitoring into the future. But we're in it for the long haul. We have to still figure out exactly what this tool is and how to best apply it. So the answer is not yet.
Right, but it's coming.
But it's coming.
The next question from Kristen, and I'm not going to butcher your last name, Kristen. How can we access or see the data from the 2020 study once it's finalized?
So if that is the study on the 2 locations within the IRL versus the Sebastian Inlet, that's already published. That was published in the journal Environmental DNA earlier this year. So that is already there. I'm happy to send links if people want to reach out to me. And then, but unpublished data is sort of still sitting on my desktop or on our servers. But all the published data is available, and that includes the review papers and that first paper that was published out of the Melbourne area. And we have another paper that's coming out where we're testing different primer sets to determine which fish primers work best for our communities. And that's coming out hopefully early 2022. So that's a whole other data set that we've worked. That was in the Mosquito Lagoon.
Dr. Gaither, how could people get in touch with you?
So my email is michelle.gaither. So Michelle 2 L's .gaither @ucf.edu.
Or you can find her by going to UCF Coastal and
Yes.
clicking her that way on the
Yes,
on the
yes.
webpage.
Yes.
Other questions? Do people have other questions? I have another question to fill in a little time. At one point during my illustrious graduate career, I got to collect samples from sewers. That was fun. And we had to treat them very specifically. We had to get them on ice in a certain amount of time. I'm just wondering, with eDNA and these water samples, was there any sort of special treatment, or is that also one of the reasons that it's just going to be so awesome?
Yeah. So yes, so DNA degrades quite quickly. And so as soon as we take that water sample, we put it on ice. So we bring these big coolers out into the field with us. Ideally, we would filter immediately, but it's impractical to do that because filtering can take anywhere from 30 minutes to 2 hours for a single sample or a set of samples, and it's just impractical to do that in the field. So we have to take it back to the lab. So that means we carry these big coolers with us. We put them on ice, and then we take them to try to prevent the DNA from degrading. There's other chemicals that have been tested. People have put ethanol into their water. That seems, it's a big use of ethanol. Other people have put other kind of chemical agents in there. but ultimately it seems as if we do pretty good by just keeping our samples on ice and then filtering them as soon as we can. 6 hours. We try to get them filtered within 6 hours.
Well, that's, at least ice is relatively inexpensive.
Ice is inexpensive, and you can get, we have a project in Costa Rica where we're working with folks doing camera drops, looking for elasmobranchs. And, you know, we often have to go a couple hours offshore. And so, you know, but thank goodness for ice. You can get ice anywhere.
Right. Do we oh, yes, here we go. A question has just come in from Catherine Lowen. Is this kind of trivial slash this is a kind of trivial slash experimental question. Sorry for misreading that. But do you think that the freezing and thawing of DNA can or has impacted your data?
So that is a very good question. In fact, we ran an experiment just on that. And indeed, when you freeze, the freeze-thaw process will degrade DNA. And we find that we've done multiple freeze-thaw experiments on the same sample just to see how it impacts. And about each freeze-thaw, we lose about half of our DNA. Now, so we don't freeze-thaw them. That's one of our laboratory our field protocols. Is we collect the water, put it immediately on ice, and then never freeze it and take it back. If we have to freeze it, then everything has to be frozen. It all has to be treated the same. So if we've had some sort of error, you really don't want to make that mistake in the middle of an experiment. We try to avoid freezing at all costs, just because it will impact results. What happens is you break that DNA up. You have DNA that's already fairly small. eDNA by definition is degraded DNA typically. You know, you might have whole cells that are shed, but those shells are going to break down. Any eDNA that is not bound to some kind of particle material is going to be available to break down. And so by nature, eDNA is fragmented already. So any freeze thaw process is just going to exacerbate that. So we do our best to keep them cold and get them back and filter them as soon as possible. Once they're in the filters and they're in their buffer, then they're stable. Then the DNA is stable.
Awesome. Stephen Hall asks, how about the effects of fish breeding seasons in addition to climate seasons?
So certainly there's going to be a seasonal impact. We know this from the FWC work. So we know that fish move in and out. So there is a seasonality to what we expect to find. There certainly might be, that's what we talked about, there might be a seasonality too to how well DNA is actually retained. Because as water temperatures increase, bacterial activity is going to increase and DNA is going to be degraded faster. So theoretically, that's going to have an impact. Spawning seasons are interesting because you have so many gametes in the water at the time. So that actually is a load of extra DNA. And extra DNA specific to that species. So if you're trying to detect other species, a lot of DNA from one particular species, which may not be your target species, can actually blow out the assays for the other stuff. So yeah, that would definitely need to be taken into consideration depending upon what your question was.
All right, Jennifer Chalky again with a question about the future. Is there an area you want to use eDNA but haven't been able to yet?
I've always been hoping to get to Antarctica, but resources not there. That for me, I'm a Florida researcher. So, that survey that we did last year in 2020, I really would like to repeat and make it a seasonal survey. The problem is it was a massive amount of effort. It took 3 research teams probably a full month to be able to do those collections across the state. And so I would love to repeat that. We did those in summer, and I would really like to do that at least in the winter, spring. So it's not new sampling; it's just more sampling. So yeah, there's always new. What the springs have. This would be interesting to do in the springs, to look at again seasonality and movement of populations. That would be kind of cool. There's so many different questions that can be addressed using these techniques. People are using them in caves now to look at cave flora and fauna. People are using them in the deep oceans. That's an exciting area where that this kind of technology can really be useful. So there's the possibilities are really endless. So yes, I have a list.
I don't hear anything about sewers, so that's an unexplored, area. I'm just kidding.
Yeah, no, that is you're right. You didn't hear anything about sewers from me.
All right. We do have one more. And then this will be the last question. Rob Persons says, gametes during spawning season that are fleeting, they die or are eaten. Seems like it would improperly skew your biomass studies. Do you try to avoid collecting samples during spawning seasons?
Yeah, that's a good point. And I think the overarching point of that is that you need to understand your system before you sample. So if you're sampling, you need to know that spawning is happening by some other method, by either traditional knowledge or FWC data or visual surveys or something. Because if you were to sample during a spawning event, clearly that would mess up if you were trying to do biodiversity, or if you're trying to do biomass estimates, that would skew it significantly. So you need to know your system. And if you were targeting, a grouper that is a mass spawner, you would hopefully know where their spawning aggregations are, and then you would sample there, maybe with taking that into account, or you would have to sample before the spawning events. So yes, that would definitely skew biomass estimates for sure.
All right. Rob says, thanks. Very good presentation. We have another thanks. I appreciate your work redefining eDNA. There's a question from Mary Grace about someone needing a signature. That has nothing to do with Dr. Gaither, but someone needs a signature showing that they were here. I would like to say thank you very much, Dr. Gaither, for being here today and sharing your knowledge with us. Thank you everyone for attending. We hope you enjoyed this presentation and all of the presentations in this series. This is the final presentation of the series. Stay tuned for upcoming coastal events in spring 2020 by visiting the UCF Coastal website at coastal.ucf.edu. Thanks a lot, Dr. Gaither.
Thank you.